see my screen now?
Yes.
Okay, I cannot see you anymore, but if there are any questions, just let me know. Yeah, thank you, Leonie, for this really nice introduction and also for inviting me to give this talk, which is indeed about my PhD studies, where I focused on the relation between gut microbiota and feather pecking, which I finished actually, yeah, almost 2.5 years ago. So I would like to start actually, well, this is a very abstract poultry farm or laying hen farm. And if we are in such a farm, you may see some chickens that are laying an egg. Others might forage in the litter. But there are also chickens that are very interested in the feathers of their conspecifics, and that might even start to pull or peck at those feathers, which then creates a reaction in the receiver. So this is what we call feather pecking. But what actually causes this damaging behavior in laying hens? So we know that that feather pecking can cause mortality of victims and causes also feather damage and tissue damage. So this behavior not only reduces animal welfare, but also productivity. So in Europe, we previously had the conventional cages or battery cages where laying hens were housed in small groups. And there you can imagine that a feather pecker cannot inflict a lot of damage because the group is relatively small. But from 2012 onwards, there is a ban on these conventional cages, at least in Europe, which means that laying hens are housed in large groups where a feather pecker can, of course, inflict more damage because they have more victims they can peck at. Then we still had beak trimming, where the sharp part of the beak is cut off to limit the damage that is done by feather pecking. But also from 2018 onwards, there's a ban on beak trimming. So now hens have sharp beaks with which they can inflict more damage upon their victims. So actually this requires alternative solutions to control or maybe even prevent feather pecking. So over decades, there's been already research on feather pecking. So we know that a lot of factors are involved in the development of these behaviors. It's multifactorial. So for example, in larger groups, you often see more feather pecking and more feather damage. Like I said previously, if you have one feather pecker and it is in a large group, it can inflict or it can cause multiple victims. Then important is also the feed. So if you provide a high fiber diet, you usually see that there is less feather pecking. The access to litter is very important. If laying hens don't have access to litter, you will definitely see more feather pecking. And also light intensity. High light intensities are often related to more feather pecking and more feather damage. So these are all external factors that might influence feather pecking, but there are also internal factors. So for example, anxiety or fearfulness, previous studies have shown that more fearful individuals are more likely to become a feather pecker. Stress is also related to feather pecking. So high stress, you often see more feather pecking. And serotonin, which is a neurotransmitter in the brain. Here we see also relations where high levels of feather pecking are often related to low serotonin levels. And there are even some studies that show relation with the immune system, where an immune challenge actually caused hens to start feather pecking. So we know that feather pecking is multifactorial, And although over decades there's been research on this topic, there is not one solution to prevent or control it. But there is some hopeful alternative. So the gut-brain axis has actually received a lot of attention in most recent years, which is a crosstalk between the gut and the brain. And this occurs via several routes. So you have a direct route, which is the neural route via the vagus nerve, where there is direct communication between the gut and the brain. Then you have the immunological route where bacteria can activate immune cells to produce cytokines, which then signal to the brain and alter behavior there. Or the metabolic route. So, for example, bacteria can influence tryptophan metabolism, and tryptophan is a precursor of serotonin. They can produce short chain fatty acids or even neurotransmitters like serotonin themselves, which then signal to the brain. And then we also have a stress route via the hypothalamus pituitary adrenal axis, through which the brain can actually influence the gut microbiota. So maybe you already see some similarities with some factors that can influence feather pecking. And actually, there's been quite some studies in rodents that link the gut microbiota to behavior, where most studies are performed on germ-free rodents, or where they provided, for example, anti-prey or probiotics. And there are even some studies that look at microbiota transplantation, so transplanting microbiota from one individual to another to see whether this affects behavior. So what is interesting is that these studies in rodents show that gut microbiota can actually influence anxiety or fearfulness, stress, serotonin, and also the immune system. And to give you one example, if you have a mouse that is fearful and less active, so fearful, it sticks to the side of an enclosure and it's not so active, and you give it a probiotic, will become more active and less fearful. So it will stick more to the center of an enclosure. So similar studies have been performed in poultry, where most focus actually on quails and chickens. And these studies also indicate that the gut microbiota can influence fearfulness, stress, serotonin, and the immune system in chickens as well. So this is a very interesting avenue of research to pursue further. And this is even more related to feather pecking, because there's been one study that looked at the microbial metabolites, so the products of the gut microbiota. And what they found was that the high and low feather pecking selection lines, so they selected laying hens on high feather pecking frequency and low feather pecking frequency. And what they saw is that these lines differed in these microbial metabolites. So that could be a good indication that their gut microbiota is also different, and that maybe the gut microbiota might play a role in feather pecking. So this was actually the aim of my PhD, to identify the effects of gut microbiota on feather pecking. And to do this, we started by with the question whether these feather pecking selection lines actually differ in gut microbiota composition. So to do this, we characterize these lines with regard to their behavioral and physiological characteristics. So just to give you an indication of the types of behavioral tests we did during these experiments. So here you can see a novel object test, where you place a novel object in a home pen, and then you record the latency for the first chick to approach the novel object, and also the number of chicks that approach it. And we did this at several ages with each time a new novel object. And this is to assess fearfulness and exploration. We also performed so-called isolation tests where you isolate chick or laying hen. So this is the bucket test, we call it, and this is the more adult variation to this, the open field test, where you record the latency to vocalize or the number of vocalizations and also the activity, so the number of steps or flight attempts. And this is also to assess fear, but also social reinstatement and exploration. Then we performed a tonic immobility test, which is actually considered the test for innate fearfulness in poultry, where you record the latency to self-righting. So actually the duration of tonic immobility is a clear indication. And then we had a manual restraint test where you restrain the bird for 5 minutes and score the latency to struggle and vocalize, and also the number of vocalizations and struggles. So these hens were tested in quite some behavioral tests. And just to quickly show you how these high and low feather pecking lines actually differ with regard to their behavior. So what we noticed was that if you look at the high feather pecking line, the chicks would approach a novel object sooner and more chicks would approach it. They would vocalize sooner in the bucket test and also more. They would also vocalize sooner in the open field test. And they would have a shorter tonic immobility duration, so they would the latency to self-righting was shorter, and they would also vocalize sooner in the main restraint test. So this is actually very consistent across studies. So also earlier studies from Marjolein Kops, we see that these high feather pecking birds, they respond more actively to all these types of behavioral tests, and it could indicate reduced fearfulness. So then we also sampled the gut microbiota, and this was done at 30 weeks of age. So actually we looked at both the gut wall, so to say, and also the gut content. But for now, I would like to focus on the gut content. So we sampled the ileum, the cecum, and the colon, which you can see indicated here. So it's actually the hind gut, so to say. And then the microbiota was determined by 16S rRNA gene sequencing. So if we then look at the differences between these two lines, and this is actually very interesting, it's consistent across studies. We see that the high feather pecking line has a higher relative abundance of Clostridiales, and the low feather pecking line has a higher abundance of Lactobacillus. And they are not, they differ in their overall composition. So as I said, we actually identified the gut microbiota in the luminal content, but a previous study looked at cecal droppings and they found exact same differences. So this is very consistent results among different studies. So we see that just to wrap up this first part where we identified actually how these genetic lines differ with regard to their behavior and their physiology, we see that these high feather pecking birds respond more active to all these kinds of behavioral tests, and that could indicate reduced fearfulness. While previously actually feather pecking has been related to increased fearfulness. So that is interesting. But the more active responses might be related to a higher activity level in general, because actually Jürgen Kjaer previously showed that these high feather pecking birds have higher locomotor activity in their home pen. And he suggested that that might relate to an altered intrinsic motivation to be active, which then leads to more locomotion and also these more active behavioral responses in our fear tests. So they might not be necessarily less fearful, but they have just a more active way of coping with things. Then we see that there are differences between these lines with regard to their microbiota, but microbiota composition is of course influenced by many factors, of which genotype and diet are relatively important. But as we see these consistent findings across studies, we think that there is a strong influence of genotype, although differences might also arise because of feather eating, as high feather pecking birds have been shown to ingest more feathers. And if feathers are provided in the diet, you actually see altered microbiota composition with an increase in Clostridia, which is similar to what we see in differences between the high and the low feather pecking line. So yeah, it might be the genotype alone, but could also be a combination with this feather eating that actually creates these differences between the high and low feather pecking line. So now we could answer our first question. Do feather pecking selection lines differ in gut microbiota? Yes, they do. And then we had a follow-up question. Does gut microbiota actually influence the development of feather pecking? So could it actually be a factor not only related, but also influencing feather pecking? So to do this, we actually used this microbiota transplantation setup, which was previously done in rodents and quails, where you transplant microbiota from one individual to another. And often what is seen is that the behavior is adopted from the donor line. So for example, if you have a fearful mouse and you give that microbiota to another mouse, that mouse will also be more fearful compared to a control. So this is also what we hypothesized for our study, so that if we would give the microbiota from a high feather pecker to a genetically low feather pecker, that it would then become a high feather pecker. So to do this, we created these microbiota transplantation pools, which were actually a mixture of the luminal content from our first study. And these are the compositions of those pools. So on the left, you see the high feather pecking pool, and on the right is the low feather pecking pool. And here you can clearly see the differences between the two. Here with the high feather pecking line having a higher abundance of Clostridiales, and in the low feather pecking line almost 2/3 is Lactobacillus. So those are clear differences. So we made these pools, and then so we had 3 treatments. So we either had the high feather pecking pool, a saline or control treatment, and the low feather pecking pool. And these treatments were then given to either high feather pecking chicks or low feather pecking chicks. And we gave the first treatment within 6 hours post-hatch, as we know that the gut is rapidly developing after chicks hatch from the egg. And also during the first 2 weeks of age, because then the brain and immune system are still developing. So we chose this time period to have maximum impact on the gut microbiota and the development of chicks. So then we also performed the same behavioral tests as we did in the first experiment. So this is again the novel object test. So just to remind you that we only gave the treatment during the first 2 weeks of age, but we also analyzed the behavior after the treatment. So the first novel object test was during treatment, and the second was at 5 weeks of age. the so-called bucket test was also within the treatment period, but then we performed the open field test at 13 weeks of age. The tonic immobility test was done at 9 weeks of age, and then the main restraint test at 15 weeks of age. And what is interesting here is that actually during the treatment, so within the first 2 weeks of age or immediate effects, we see effects only on high feather pecking chicks that received high feather pecking microbiota. So what we saw was that these chicks would approach a novel object sooner and more chicks would approach it. And they would also vocalize sooner in the isolation test. So this again could indicate reduced fearfulness in these chicks receiving microbiota from their own line. Then after treatment, this is from 13 weeks of age onwards, or actually long-term effects, we only see effects in low feather pecking chicks receiving low feather pecking microbiota. And here we see that these chicks would step sooner and vocalize sooner in the open field test. And they would also vocalize sooner in the main restraint test. So also here we see that they would have these more active responses, which could indicate reduced fearfulness. Then we also sampled actually the microbiota of the recipients to see whether our microbiota treatment actually altered the gut microbiota. And to do this, we sampled the gut content again of the ileum, cecum, and colon. And this was done at 5 days of age and at the end of the treatment period, so at 2 weeks of age. So here we saw that there actually weren't any differences between the chicks that received low feather pecking or high feather pecking microbiota, but the control treatment, so the saline treatment, did differ from chicks that received either the low or high feather pecking microbiota. And this was seen at 5 days of age and also at 2 weeks of age. So even though we didn't see differences between chicks receiving high or low feather pecking microbiota, we did see differences with receiving adult microbiota, yes or no. And it should, this is only seen in the cecum, so actually the hindgut, would say. So we saw limited effects on recipients' microbiota composition, which might be caused because these pools consisted of adult microbiota, which might not be able to colonize and remain in the gut, as the gut is still developing and there's a rapid succession of gut microbiota, especially just after hatching, after first feeding. But we do see effects on these behavioral responses, and that might be explained by that we gave the transplantation when brain and immune system were still developing. So we know that microbiota actually affects the development of these systems in rodents. For example, it alters the morphology of the amygdala, which is the part of the brain that is involved in the regulation of anxiety and fear. And also it influences the expression of the brain-derived neurotrophic factor, which is a gene involved in synaptic plasticity in the amygdala. So just to remind, these are studies in rodents, not in poultry, but still similar, similar actions could cause the gut microbiota to alter behavior responses in poultry. So if I go back to our question, actually, do recipients adopt the behavior of donors? So from the first study, we saw that high feather pecking birds had more active behavior responses. And during the treatment, we see that these high feather pecking chicks receiving high feather pecking microbiota indeed adopt the behavior of donors. But then after treatment, we see that low feather pecking chicks that received low feather pecking microbiota became more active. So they do not adopt the behavior of donors because low feather pecking birds are less active actually. So what might be an explanation that we see these immediate effects in the high feather pecking line is that we also see that the high feather pecking line is has a more responsive immune system. So they might respond more strongly to our gut microbiota given during the first 2 weeks, which then results in the production of cytokines that can directly alter serotonergic and dopaminergic neurotransmission in the brain, and which then leads to direct alteration of these behavioral responses. But we see the long-term effects on behavior in the low fat pecking line. So what might be explanation there? So actually the low fat pecking microbiota had a higher relative abundance of lactobacillus. And we know that lactobacillus actually increases activity and reduces anxiety in rodents. So this might be why the low feather pecking chicks that receive low feather pecking microbiota showed these more active behavior responses. Might be because they received this amount of lactobacillus. However, here, it should be noted that we did not actually find a difference in microbiota composition of the recipients. So we did not see this increase in lactobacillus for chicks receiving low feather pecking microbiota. Still, what is interesting is that we see this effect of homologous transplantation. So transplanting microbiota from the same genotype to the genetically similar chicks. So from high feather pecking microbiota to high feather pecking chicks, and from low feather pecking microbiota to low feather pecking chicks. And that in both we see that they have more active responses that could indicate reduced fearfulness. But yeah, so like I said previously, feather pecking has been related to increased fearfulness. So this might be a way to reduce fearfulness and reduce feather pecking. Although it should be noted that we didn't actually find differences in tonic immobility duration, which is the measure for innate fearfulness. And as I showed previously, feather pecking in these high and low feather pecking selection lines was related to reduced fearfulness. So still to conclude, we see that the effects of microbiota transplantation are genotype dependent, where we see immediate effects in the high feather pecking line and birds adopting the behavior of donors, and long-term effects in the low feather pecking line with birds not adopting the behavior of donors. But still, a microbiota transplantation can affect these behavioral responses, where we see that homologous transplantation actually reduces fearfulness. And in that way, microbiota could influence the development of feather pecking. I would just like to show you some recent findings with regard to feather pecking and the gut microbiota. So these are two studies from Claire Minnes from the University of Guelph. And she looked at supplementing laying hens with probiotics. So here it was specifically Lactobacillus rhamnosus. And what they saw was that the laying hens supplemented with this probiotic actually prevented an increase in feather pecking after a stressor. Also had an effect on the immune system. So they saw an increase in regulatory T cell population. And it limited cecal microbiota dysbiosis. And dysbiosis can also be related to stress. And in another study, they also noticed that supplementing with a probiotic increased plasma tryptophan and turnover. So tryptophan is again the precursor of serotonin. So I think this is a really nice kind of follow-up study that actually showed that with the probiotic and altering the gut microbiota, you can do a lot and might even be able to reduce or control feather pecking. Then I would just like to briefly highlight some projects we have within Wageningen Livestock Research that are related to this topic, so relating the gut microbiota to behavior. And one project actually looked at associations between the early rearing environment, so providing a dark brooder, yes or no. And in the picture below, you can see a dark brooder, which is actually a safe area for the chicks to rest under, and it's also so it provides a dark and warm area. And in this study, you looked at associations between having a dark brooder, yes or no, and the gut microbiota, behavior, and welfare of broilers. And what was found was that providing a dark brooder actually had positive effect on welfare and also affected the microbiota diversity. Then a second study, here we actually tried to reduce endotoxin concentration in the litter via the modulation of the gut microbiota through feed interventions. And here we saw that adding medium chain fatty acids affected the microbiota diversity and behavior. So for both these studies, please do not hesitate to contact me if you have any questions, or my colleague Ingrid de Jong. So with that, I would, well, these are just small take-home messages, and I would like to thank you for your attention and thank you for inviting me to give this talk. And if there are any questions, I'll be happy to answer them.
Thank you very much, Jerine. There are a bunch of questions from the audience, so we can go over them.
Should I stop sharing?
You can keep sharing. Maybe
Okay.
you want to go to slides to reference them. And I'll also post your email address in the chat if people have direct questions. Excuse me. So the first question, and I think maybe the answer was already given in your talk a little bit, but still I wanted to ask. Could you explain how in a single flock of birds coming from the same incubator, from the same hatchery, how they could get different microflora?
How they would get different microflora? Yeah, well, of course, individuals differ. Individuals differ, so they will behave differently, and maybe some stick to a certain area of a house, and others are exploring more. So then you could create these differences with the more explorative birds pecking at different types of litter, or pecking at different, I would say, droppings from other individuals, and this could influence their microbiota in such a way. And then, of course, also an important part is the genotype. So all these individuals are not identical. So that could also influence their gut microbiota composition.
All right, thank you. Another question, are behavior and gut microbiota differences between high and low feather peckers consistent across different breeds or strains?
So for the studies, most of the studies that I mentioned looked at these high and low feather pecking selection lines, and they are actually specifically selected for this behavior. But of course, you also have, for example, white and brown laying hens that differ a lot in their behavior. For example, white laying hens are thought to be more fearful and often also show more feather pecking, while the brown ones are are maybe more influenced by environmental factors, so to say. So yeah, wait, I forgot your question.
So the question is whether, I guess, the differences between the feather peckers and the nut peckers are consistent across different strains.
Yeah, so that's difficult to answer because even within these high and low feather pecking selection lines, you can have feather peckers and non-peckers. And I actually also looked at the specific phenotype, but yeah, I'm not sure about other breeds. I cannot tell you there, but what we do see is that these lines already differ with regard to other breeds. If you see that feather pecking is related to these more active behavior responses and the reduced fearfulness, while for the more commercial breeds, so to say, you see that feather pecking is related to being more fearful. So yeah, I I think it's tricky. I cannot give you an answer.
Could you say which strain you're using or you used in your research, or is that a confidential information?
No. So these were actually white leghorn laying hens, but they are from quite some years ago because they have been selected and then maintained for quite some generations. So I don't think we can even compare them to the nowadays commercial white leghorn. Yeah.
All right. Another question related to genotype. Since genotype was implicated to exacerbate feather pecking, is there a study as regards its effect within breeds?
Yeah, so like I said, well, I hope I understand the question, but so even within the high feather pecking line, you can have feather peckers and non-peckers, so to say. So it's not that all of them become feather peckers. So there are still individual differences. And also in the low feather pecking line, we still see feather peckers. So yeah,
This is interesting.
yeah. You cannot like solely breed on non-feather pecking and then remove it, so to say. It's also difficult to select on a behavior not being there. So yeah, we see that the high feather pecking selection lines actually is a clear increase in feather pecking, but still individuals that don't show it. But then for the low feather pecking line, yeah, how long can you select for something that is not there? So that's tricky. So I don't think that genetic selection is the solution, if that's the question.
All right, so you mentioned that eating feathers impacts their microbiota. One question is, what response would you expect if a flock is fed with feather meal as part of the diet?
Yeah, so actually then they also tried this. So they gave feathers in the diet and then they saw this reduction in feather pecking. So it might also be a need for them to have a specific type of nutrients. So for example, also if you provide fiber in the diet, you see a reduction in feather pecking. And actually Alexandra Harlander looked at this as well, and she's together with Claire and Nienke van Staveren, they are still looking also at gut motility and how this affects feather pecking. So I think there's a lots of more interesting research there to see whether we can influence feather pecking in this way, maybe with the feed or a probiotic. Yeah.
Well, still a bunch of questions, so we
Yeah.
have time. Someone asked whether litter will impact gut microbiota and if there is a way to sort of control the impacts of litter.
So yeah, litter can definitely impact the gut microbiota because it's also something which they take up, of course, when they are foraging. So that could influence it, but I don't know if different types of litter cause different types of gut microbiota composition to arise. I guess it would, but I don't know how this could then be used to influence feather packing. Maybe, well, also another nice further research topic.
Yeah, I wonder if you have thoughts on how you could test the effect of litter besides, I guess, comparing it to not giving litter, but that can impact the bird in other ways too.
What do you mean?
So we know that chickens like foraging. If you don't give them access to a foraging substrate, that can impact their behavioral repertoire, anxiety maybe. So is it really the effect of litter that impacts the birds? I know it's a complicated question. Or is it having the ability to forage and showing those behaviors and sort of meeting some of those motivations?
I think with regard to litter, probably the last is most important because this is also one of the hypotheses why feather pecking arises, is that there's they laying hens have this strong motivation to forage and to search for feed, and if they cannot express this motivation, then they will kind of redirect that foraging motivation to pecking at their conspecific, so to say. But it would actually be interesting to see whether different types of litter would also affect the amount of feather pecking and whether we can somehow reduce it there. I could imagine that if the foraging or if the litter is like a foraging substrate that is preferred or that is more easy um, to forage and to motivate them to forage, that might limit feather pecking as well.
Thank you. Another question. There is evidence of a social learning component to the development of feather pecking. Has any research been done to look at sort of the social learning in addition to the effects on gut microbiota? For example, all the birds were tested in groups in the fear test. How may the group influence behavior, and what if you mix the genetic strains like the high feather pecking lines and the low feather pecking lines, and then look at behavior?
This is a good point. So indeed, we kept the lines separate. So you could imagine that there might be one high feather pecking bird that then influences all the other ones in the group. But at least for this first initial study, we didn't want to divide them because then you could also get that they influence their gut microbiota. Like I said previously, if we would have housed high and low together in one pen, they would also peck at each other's droppings, and then you will get a mixture. You could get a mixture of both. So that's why we housed them separately, actually. But this would be a very interesting study as well. We were actually thinking about mixing them to see whether you would still see these effects if they are housed together. But yeah, unfortunately for this first initial studies, we decided, okay, we have to separate them to see whether there are any effects, and if there are, then you could go on further with such research. Yeah.
Right. Another question is, how does methionine and cystine, I'm not sure how you pronounce that, levels in feed affect feather pecking? Because we know that feathers contain a lot of cystine, but at the same time, methionine is reduced to control egg size.
So I have to say that my my knowledge about these specific nutrients is not sufficient, so I cannot give an answer to this question. But
okay.
yeah, I do know that like I said already previously, that this feather eating or providing feathers in the diet would actually also reduce feather pack. So there is something there, but with regard to these specific nutrients, I cannot give an answer. Sorry.
And have you seen, I guess maybe in the more recent research that you alluded to, if there's an association between probiotic use and environmental enrichment, for instance, pecking objects, if that helps to reduce feather pecking behavior?
Well, of course, providing enrichment to which these laying hens can show their foraging behavior or motivation is always helpful, I think. But I cannot say how that relates to the gut microbiota. For this dark brooder experiment, we actually specifically selected a treatment that wasn't peckable, so to say, because they might also influence the gut microbiota. So we wanted to provide an enrichment in early life without it being able to affect the gut microbiota to see what the effects are. So that's difficult to then disentangle. But yeah, we are still thinking of also looking at these types of enrichment. But you can then never say whether it's the enrichment that is influencing the microbiota just because it's there or because it's also being eaten, so to say.
Yeah. One audience member is wondering if there are any similar studies in swine or piglets, and I'm assuming them related to other injurious behavior like maybe tail biting. Do you have any suggestions of researchers maybe that you know of that are looking into this?
Yeah, so I know there is actually, so tail biting is very comparable to feather pecking in a sense. And I know there are people that would like to study this. And I know that, for example, Anushka Middelkoop actually also looked at the relation between the gut microbiota and behavior in pigs. So definitely look for her studies. And then I think also Frank Duitens actually wanted to look at this, but I'm not sure whether he's actually doing this already or that it's like in progress.
All right, thank you. There are some questions from the panel, us, about your behavioral tests. How did you make decisions about when you performed your behavioral tests? Like the novel object test was at a quite a young age when they were less than 2 weeks old, and the open field test when they were 13 weeks old.
Yeah, so this is both. So what we tried to do was do a test at a young age and then also at a more adult age. So that is why, for example, the bucket test was kind of the early version of the open field test. So that is what we try to do. And then also there are some practical aspects because young chicks, of course, have to be kept at quite high temperatures. So that is why we performed those tests in the home pen to so that you don't have to isolate them. And then the bucket test was at a bit older age, so that you could actually isolate them. So those two factors were involved in making this decision.
Thank you. And I was wondering, you were talking about the bucket test and that birds or the high feather pecking line focalize sooner. Could that be, and which was interpreted as a less fearful response? Could it be the opposite, where they may just be more fearful depending on the type of focalization? Was that looked into?
Yeah, so this we didn't look into this, so this would be very interesting. Just it was focalizations in general, but what it also says is well, it's difficult with these tests because it can be fear, it can be social reinstatement as well. And I think these vocalizations, they are of course also a type of social reinstatement. So them vocalizing more, you could also say they're more socially oriented. Maybe not in the good way, but yeah. So I I actually don't prefer to say that they are less fearful. I think they are just more, they have this more active way of coping with any situation, which is why they vocalize sooner and more. And that might also be why they start to feather pack. Because it's just their active way of coping with their situation.
Yeah. Okay. There are some questions about the methods. Were microbiome analyses conducted at all ages, or like multiple ages post hatch, or yeah, I guess that's the question. When?
Yeah, so for this last study where we transplanted the microbiota, we actually looked at 5 days of age and 2 weeks of age, where the 2 weeks of age was at the end of the treatment. But we didn't look at very long-term differences in gut microbiota because we would because we didn't find clear differences at 2 weeks of age. We expected that, for example, at 15 weeks of age, we also wouldn't see any differences. So that's why we focused on those kind of early ages for sampling. Yeah.
And were metabolomics considered at all during your experiments?
Yes, yeah, we thought about this too. But at some point you have to you have to focus on particular aspects, and because we already included, so I actually, apart from the behavior, I also looked at the immune system, for example, and serotonin and stress. So we kind of limited ourselves in that aspect, that we only looked at microbiota composition and not metabolomics. But this is would be very interesting to also see whether these lines actually differ with regard to metabolomics. Yeah.
All right, and a final question, maybe very theoretical, but you, although you haven't showed it or found it in poultry, that microbiome sort of has a causal relationship or influences feather packing. What do you think would the theoretical benefit be for the microbes to sort of influence the bird to feather pack?
Yeah, that's interesting. This is actually also something, if you look at these germ-free mice. Well, of course, if for the gut microbiota, it would be beneficial for them to kind of explore their environment more, or if these gut microbiota specifically need certain substrates to grow and develop within the gut, then of course it would be beneficial for them to direct the birds to their feathers. But that's a very specific specific example, and but I can imagine that if you have microbiota that would make animals more explorative, that then maybe they would have different substrates, or the other way around, if they would make them less explorative because in a certain area there is nutrients or substances that they need to grow, that would be beneficial for them not to make those animals more fearful and less explorative, so to say. So in that sense, it would be beneficial for them.
Yeah, no, I think that's a good answer.
But that's really theoretical, yeah.
Yeah, maybe we'll find out in a few years.
Yeah.
Well, again,